research paper highlights the anorectic effect of neuropeptide AF as it is associated with satiety-related hypothalamic nuclei. This presentation is by Natalie, Morrow, Devyn, Zewdie, and Megan Daniels.
Food intake is regulated by various neurochemicals. Some suppress hunger and are known as anorexigenic, while others promote hunger and are known as orexigenic. This is a very complex topic, and the exact mechanism of how this works isn't entirely understood because not all neurochemicals that can play part in this process have been discovered. Appetite biologists have been studying a particular set of neurochemicals known as the R-famides. Neuropeptide AF is an R-famide. NPAF has been found in the rat, mouse, and human brain. It is located in the hypothalamus near regions that involve satiety and hunger. Researchers in this experiment hypothesized that NPAF may reduce hunger since it and neuropeptide FF, another anorexigenic factor, share a common precursor mRNA and are located in the same regions of the brain. This is terminology that will be used throughout the presentation. Cobb 500 chicks aged from 30 to 34 weeks old were used in this study. For each experiment, chicks from the same hatch were used, but across all 4 experiments, different hatches were used. So in experiment 1, there were 30 chicks. Those 30 chicks were just used for experiment 1. Then a different hatch was selected for each successive experiment. So different chicks were used. The room was kept at 30 degrees. The experiments were conducted between 12 p.m. and 4 p.m. Chicks were placed in a device to keep their heads still so freehand injections could be performed. The target of these injections was the left lateral ventricle, and the needle was held in the non-anesthetized chick for 5 seconds to reduce backflow. All treatments were assigned at random. In experiment 1, part 1, the chicks fasted for 3 hours and were randomly assigned to receive between 0 and 16 animals of neuropeptide AF by ICV injection. After injection, the chicks returned to the cages and food intake was monitored every 30 minutes for 3 hours. In part 2 of experiment 1, trunk blood was collected from the chicks right after the 180-minute food intake reading, and the blood glucose was determined. Now in experiment 2, parts 1 and 2 were identical with experiment 1. The only difference in experiment 2, part 1 is that food was restricted post-injection for the whole 180-minute period for the chicks. Experiment 3, the chicks fasted for 3 hours and were randomly assigned to either receive a vehicle or 8 animals of NPAF by ICV injection. 30 minutes post-injection, they were anesthetized and then decapitated. After decapitation, slices of the brain were taken using a grafting. Certain regions of the brain were collected, and the number of reactive cells which showed presence of C-pulse was averaged between all the brains. Experiment 4 was conducted to study the behavioral effects of the chickens post-injection. The chicks were kept in individual cages. They could see but not hear their neighboring chicks. The chicks either received 0 nanomals or 8 nanomals of NPAF by ICV injection after fasting.
Now looking at the results for part 1 with the blood glucose concentration, the chicks that were treated with 4 nanomoles of NPAF did not have a reduction in their food intake, but those chicks treated with 8 nanomoles and 16 nanomoles did have a reduction in their food intake, and this stayed effective for around 180 minutes post-injection. In the previous study with NPFF, when delivered at higher doses, 8.32 nanomoles to 16.6 nanomoles had a reduction in food intake only up to 120 minutes post-injection. Another study with LP-LRF amine was used, but the effect only lasted 40 minutes post-injection. They believe that this is due to the peptide plane. The NPAF also reduced water intake, which contradicts other RF amines peptides, as NPFF and NPVF had no effect on water intake, where NPAF did. Part 2 of this experiment, a glucose concentrations were seen to have been affected by the NPAF injection. This decrease in concentration is believed to be because of the decrease of carbohydrates available in the chick due to its decreased food intake. But only the 8-animal and the 16-animal injections of NPAF proved to have a decrease in food intake, and yet the 4-animal had a 26% decrease in glucose concentration. Based on this, it is believed that the NPAF stimulates the carbohydrate utilization. For experiment 2, part 1, we are looking at the same experiment but with the absence of food. The chicks treated with 8 animals and 16 animals showed a reduction of water intake. When looking at part 2 of this experiment, the blood glucose levels were not affected. In experiment 1, it was thought that NPAF stimulated the carbohydrate utilization. It is now evident that it does not, as the concentration levels did not change. After these 2 experiments, NPAF is believed to affect glucose independently of food intake, but it is dependent on the feeding conditions. Experiment 3, CFOS was set as the indicator for neuronal activation. There was an increase in CFOS immunoreactive cells in DMN, PAMC, PAPC, and VMH. This suggests that all these regions are associated with NPAF-induced anorexia. All are perceptions of satiety. This is seen as a primary response, as the increase in CFOS in these regions was because of the result of increased satiety, not as an effect of food reduction. Experiment 4 is looking at the behavioral effect of NPAF. The feeding pecs and the time purging were both reduced, 15 to 30 minutes post-injection, indicating a slight lag time. The pecs are not necessarily related to food intake because the food was not consumed every time it went to pec. The pec efficiency was not affected by NPAF. There was no behavioral effect on escape attempts, defecations, nor distance moved. It did cause more deep sleep, but it was hard to tell if it was just a primary effect on behavior from the NPAF injection or just an anti-appetite response. In summary of this experiment, NPAF did cause a decrease in food and water intake when injected with 8 animals and 16 animals of NPAF. Also, the NPAF was different from other RF-amines as it had a longer lasting effect on the reduction of food. This is believed to be so as NPAF is a longer peptide in reference to NPFF and LPLRF amine that both had significantly shorter effective times. So the researchers concluded that the longer the neuropeptide was, the longer effect it had in decreasing food intake. At first, NPAF was believed to have stimulated the carbohydrate utilization, as the concentration for all injection levels of NPAF created a decrease in glucose concentration levels, but it was later found that this is dependent on food conditions, as the chicks that did not have food available to them did not have a change in their blood glucose levels. There was proven association with DMN, PAMC, PAPC, and VMH, as there was an increase in CFOS immunoreactive cells in these regions of the brain, leading the researchers to believe that these areas were responsible for satiety. The behavioral effects that were observed were an increased deep sleep and a decrease in food packs and time purged.
While this experiment plays a critical role in deciphering the mysterious world of RFMIs, and specifically NPAF, there are a few aspects of this experiment that could be revised or looked into further. The fact that the chicks all come from the same hatchery does allow for the results to remain consistent, but it makes the focus very small. I would recommend that these preliminary findings be examined on a larger scale, with the chicks coming from multiple hatcheries instead of just one to validate the conclusions. The large number of factors, such as food and water intake, behavior, glucose levels, and CFOS activity, highlight the breadth of the variables in this study. While showcasing all the effects from the injection of NPAF during the experiment, it also shows that the interactions between them are not often understood, except for the independence of food intake, which is dependent on feeding condition, and glucose homeostasis. These points of examination may want to be narrowed to provide interaction amongst these variables instead of NPAF. As mentioned in the experiment, the prolactin-releasing peptide and the gonadotrophin-inhibiting hormones cause overeating in chicks, but does the exact opposite in rats. This distinction is an important one, as these results would be hard to apply to large swaths of organisms, particularly humans. The findings of this experiment put one more piece of a very complex puzzle together in the context of satiety for humans. Discovering how the pathways work could have mass implications for the majority of people due to a better understanding of rapid weight gain in America.